Bismillahirrahmanirrahim. So the next topic is CJD. In dementia, we have studied about various types of dementia. So our next topic is CJD. That is called as Creutzfeldt-Jakob disease. So actually, this is the one of the part of prion disease. So we have got various types of prion diseases, right? So first one is CJD. The second one is Gerstmann Straussler-Schenker disease, right? The third disease is the fatal familial insomnia, and the fourth one is the sporadic fatal insomnia, right? Sporadic fatal insomnia, and the fifth one is Kuru disease, right? So these are the five types of prion disease. So we're just gonna discuss about the CJD, right? So let's talk about CJD. Let me erase these things here. Okay, so so prion proteins actually, what they does is that they deposit within the cells. Prion protein deposit within cells and have ability to corrupt the native proteins. So these are the infective agents. So these are the unique amongst infectious agents, that they are devoid of nucleic acids. Most of the infectious agents, they have nucleic acid, but these are the proteins, special type of very unique proteins that are devoid of nucleic acids, right? And what they does that they convert p prion protein PRPC, that is normal type, into PRPSC type, right? That is beta-braided sheet, and this is resistant to our protease. So our normal protease, what they does is, protease, they can cleave alpha-plated sheets only, right? So they cannot cleave beta-plated sheets. So when they cannot cleave beta-plated sheets, so this is going to deposit in our cells, right? If it deposits within our interstitium, so this is going to cause the amyloidosis. If it deposits between the neurons, so this is going to cause the Alzheimer disease. And also this is going to cause if it deposits within the cells of the brains, within the neuronal cells, so this is going to affect the other proteins, and this is going to cause the CJD, right? So let's talk about sporadic form. Sporadic form is actually there's idiopathic form, right? We don't find any cause of that. All right, so the next type is familial form. So in this form, we have got the defect in PRP gene. So when there's defect in PRP gene, so this is going to cause the prion disease. And another one is iatrogenic, like if we transplant cornea from infected person, it will cause the recipient, this is going to cause the CJD, right? So if we do the corneal transplant, or if we do the cadaveric durameter transplant, right? So if we put the depth EEG electrodes, right? So this is how this is going to spread to the recipient, right? So we have got VCJD, that is called as variant form of CJD, right? So actually this is this we can get from the ingestion of infected meat, ingestion of basically neuronal tissues. Okay, infected neuronal tissues, जो लोग को मगज़ खाते हैं एनिमल्स का, if there is any infected animal, so if some person eat that, so he is gonna get the prion disease, he's gonna get the CJD, right? So actually what happens that if we talk about the triad of the human prion disease, so in triad of human prion disease, so we'll have the cortical spongiform pattern changes, and we'll have the neuronal cell loss, and there will be gliosis. So if I just draw the triad here, so this is the human prion disease. So this is actually the histopathological triad. So there will be first thing is that there will be neuronal cell loss, right? And there will be gliosis, and there will be cortical spongiform form change. So this is the triad for the human prion disease. So let's talk about the features of this prion disease. Before that, let me go back to our topic in a small concept in the that I have discussed in the Alzheimer disease section. So let me talk about that. So we have got the types. If we talk about the types of protein foldings, so we have got the alpha plated folding, right? Another one is beta plated folding. So our human protease can only cleave these alpha plated proteins, right? So human protease can cleave only alpha plated proteins. They cannot cleave the beta plated proteins, right? So um, develop if we develop the excessive amount of beta plated proteins, so this is gonna deposit in the body organs. If it deposit in the interstitium, this is gonna cause the amyloidosis. If it deposits within the cells, so this is gonna cause the CJD, right? And if it deposits within the neurons, so if it deposits between the neurons, so this is gonna interfere the activity of the neurons, so this is gonna cause the Alzheimer disease. So, and how we can get this how we can get this beta plated protein is because of the that's we can get from the chronic inflammation, right? Like from WBCs, they convert this alpha plated into beta plated form. So that's how we can get the Alzheimer disease. We can get the amyloidosis, we can get the CJD, right? So this was a little concept in the beta plated protein formation. So let's come back to our discussion. So we have discussed about the four variants of CJD, right? And now let's talk about what are the clinical features of this disease. So if we can see this is one of the cause of dementia. If patient is having dementia with myoclonus, right, with myoclonus and visual disturbances like vertical gaze palsy, so we're going to suspect the patient is having this CJD, that is Creutzfeldt-Jakob disease. So we can go for CSF electrophoresis. We can do the olfactory nerve and tonsillar biopsy. So it is thought that the prion disease affect the reticuloendothelial cells. So if we get the tonsillar biopsy, so we can get those infected proteins from there. And what we can do, another thing here is that we can go for the EEG, right? So in EEG we will have the periodic sharp wave complexes, right? So we have another type of complexes that is called as PLED, periodic lateralized epileptiform discharges, that we're going to see in the patients with the herpes encephalitis, right? So we're going to see this in herpes encephalitis, and that is going to affect the temporal lobe. So we don't have any treatment for this disease. So that's all for CJD, right? So we just need to memorize this thing that if the patient presents with the myoclonus and dementia and myoclonus, and patient is having visual disturbances, so we're gonna think that patient is having the CJD. So we're gonna also ask about the history, if you got the if you got any corneal transplant or dural transplant, right? If we ingested any brain tissue, right? So that's all about CJD. So let's move on to our next topic, that is called as normal pressure hydrocephalus. All right, so let's talk about the normal pressure hydrocephalus. So in normal pressure hydrocephalus, there is actually episodic rise in intracranial pressure, and that mostly happens in the night, and this mostly happens in elderly people, right? So this mostly happen in elderly people. So this is one of the cause of we can treat this dementia. Sometime we can treat this, and so what are the features? What happens actually when there's increased pressure, when there's raised ICP? So what it gonna does that this is gonna distort the corona radiata fibers, right? So that's how we're gonna get these manifestations. Like patient will be having dementia, right? So ignore the spells of that. So dementia, patient is going to have the urinary incontinence, and patient is going to have the gait disturbances, and that is called as magnetic gait, right? So this is reversible. This can be reversible if we place the shunt in these patients, like that is called as ventriculoperitoneal shunt. So this is a just treatment for that. So if patient present with the dementia and myoclonus and vertical gait palsy, so we're going to think about patient is having VCJD, right, or CJD, Creutzfeldt-Jakob disease. If patient is having dementia and urinary incontinence and gait disturbances, so we're gonna think about the patient is having the normal pressure hydrocephalus. So I think that's enough for today. So we're gonna move on to the next topic, that is called a stroke, right? So that's all for today. Thanks for watching.